Okay, in this video, I'm going to go over chapter 7, which goes over genomes and chromosomes. This is probably one of the harder chapters in the class since we're going to go over things like DNA replication. Yeah, so it's a process. So we're going to first talk about DNA just in general, and we'll talk about how the DNA is kind of organized in the bacterial cell. But then, yeah, the main bulk of it is talking about DNA replication and the plasmids. And then at the end, we kind of just briefly go over the eukaryotic and archaeal chromosomes. So we're really focusing on bacterial chromosomes here, but we do touch upon the other types of chromosomes. And then finally, just a couple things about microbiomes, and metagenomes. Okay. So 7.1, we're just talking about genomes in general. So you should probably already know that a genome is all of the genetic information of an organism, and a chromosome is the main molecule of DNA of an organism. So as you can see here, different types of bacteria have different sizes for their chromosomes. And something that you might notice is that most bacteria have circular chromosomes. So pretty much all of them except for this one and this one, but everyone else pretty much has circular chromosomes, meaning that the DNA is not a line, it's actually wrapped around itself, like a hair tie or a rubber band. So it's a circle. Okay. And some bacteria also have these smaller pieces of DNA called plasmids. So these smaller pieces of DNA, again, are circular, and they contain non-essential genes, but are still very useful. And, we'll talk about in this chapter what these plasmids do and how they work as well. The main bulk of what we're going to talk about are the chromosomes, but the plasmids are also something that we're going to talk about. Okay. So really quickly, just the difference between bacterial and eukaryotic DNA, because you're probably familiar with DNA in the context of eukaryotes, because that's usually what they teach in, gen bio, cell bio. But bacterial DNA is going to contain mostly genes and very little non-coding DNA. So this is bacterial chromosome, and this is eukaryotic chromosome. So as you can see in the color code, the genes are in green, whereas the non-coding DNA is in purple. Non-coding meaning it's just junk DNA, it doesn't really have a function. As you can see in humans, 98% of the genome is non-coding, meaning it doesn't really code for anything. Whereas with bacteria, as you can see, a big portion of its DNA is actual useful DNA. Again, that makes sense because bacteria are trying to be very efficient, and it's a waste of time having so many non-coding DNA. It doesn't make sense for a bacteria to be holding on to that stuff. So bacteria are very efficient at making sure that their DNA is quote unquote clean, meaning that it's just the essential genes that they need and it doesn't have any junk DNA essentially. Another thing that you're going to notice is that bacterial DNA have no introns, so they don't really have introns the way that eukaryotes do. And so they don't have to do any splicing during protein synthesis. So when they convert from DNA to RNA, that RNA is ready to go. There's no splicing needed. So yeah, good for them. Okay, really quick overview of the structure of DNA and RNA. Again, this is something that you should already know from other classes, but the structure of DNA, of course, is double-stranded. These are the bases, A, T, C, G, and it runs anti-parallel. So what does that mean? It means that the 3' end and the 5' end of one strand is opposite to the other strand. So the 3 and the 5 have to do with the carbon number. So if we're looking at the ribose sugar, in this case, deoxyribose, the deoxyribose on this DNA, this is the 3 carbon, and this is the 5 carbon. This is the 3 carbon. This is the 5. So we call this the 5' end, and then this is the 3' end. And then the other strand goes in the opposite direction. So that's why we call it anti-parallel, because they're going in opposite directions. Okay. Something else to note is that the 5' end is where you would find the phosphate group. And that's going to be important when we talk about DNA, DNA replication, because you can only add a nucleotide to the 5' end, not to the 3' end, because we need that phosphate group to add another nucleotide on top. Okay. In RNA, it's a little bit more simple. It's single-stranded, so you should know that. The base is AUCG. And yeah, that's pretty much it. And there's also a 3' and a 5' end, pretty similar in structure, other than the fact that it's single-stranded and there's a U instead of a T. So okay. So now we're going to talk about the structure of, and I think this is This should be 7.3 at this point. Wait, let me double check really quick. Just kidding. No, not yet. It's still 7.2. Anyway, so now we're going to talk about kind of the way that bacteria structure their DNA. Just like how DNA in eukaryotes are structured into chromosomes that are kind of tightly wound around each other. DNA in bacteria are also packed very tightly, again, because DNA is a mess. And you have to pack it so that it doesn't just get everywhere. And so we pack it, bacteria, they pack it into this structure called the nucleoid. So the nucleoid is when the DNA is all wound up. And so the main thing that you're going to notice is that there are these large loops. So there's a big loop of DNA. The DNA is looped around each other, around itself like this, kind of like a nice little flower. And so how do these loops form? These loops form by these proteins. They're called histone-like proteins. It's called a histone-like protein because it's very similar to histones in eukaryotes, which again help with looping the DNA. But anyway, they're called histone-like proteins because they're not actually histones, but they help create these loops for the DNA. And as you can see, if you were to uncoil one of the loops, only that loop uncoils, not the whole piece of DNA, because that would be tragic if the entire piece of DNA uncoiled or unlooped. So the loops basically maintain structure by, if this uncoils, that's fine, but at least the other ones still remain coiled. Okay, the other structure that we're going to see are coils. So this is one loop, but then as you can see inside, there are these like smaller loops, and we call those coils. So this giant petal looking thing is one loop, but then all the tiny little loops inside are called supercoils. Okay, so that makes the DNA even more compact. And so how do these supercoils form? It really forms when there is a break in the DNA, or there's a change in the DNA helix structure. So for example, if we get circular DNA, and here's circular DNA, if we break it and then reseal it, but we cause a little loop around it, instead of sealing it the same way, we kind of flip it around each other, and then we reseal it from there, then it causes a loop. So I'm just going to give you an example. Here is, this is just like a ribbon, and you can attach it to one Velcro. And so if I were to break that open, right, and then instead of me putting it back the same way, I like loop it around itself first, and then I do that, then you see that it created a loop at the very end here. And so now it's not perfectly, and no matter what I do, no matter how I orient it, there will always be a little twist in there. So I've created basically a twist in my ribbon. And so that creates one supercoil. And so you could create obviously multiple supercoils, and that's what coils the DNA over and over again. Another way that supercoils happen is by just simply uncoiling the helix. So as you probably know, the DNA is structured in this helix where it's like a staircase, right? You know how a staircase is in a helix form. So what the bacteria can do is actually undo that helix and untwist the helix. And the DNA does not like that. DNA does not like to not have its helix structure, right? DNA is supposed to have this helix. That's how it's supposed to be. If you were to break open the DNA and undo one of the helixes, the DNA is now not happy. And what it will do is it will form this supercoil to kind of compensate for the lack of that twist in the helix itself. So in this lecture, I'm going to be talking about supercoils, which is like an entire loop of the DNA, versus a helix coil or a helix twist, where it's just that twist in the helix. So let me just kind of zoom in here. So as you can see, the helix, the DNA, you know how you have two strands of DNA and they kind of twist around each other in a helix? I'm going to call that a helix twist in this lecture, whereas one loop is going to be called, or one flip is going to be called a supercoil. And then a giant flip like this is going to be called a loop. So we're just thinking about it in sizes. So a single twist in the DNA is what I'm going to be calling a helix twist. And then this is going to be a supercoil. And then if you zoom out even further, this would be called a loop. Okay. All right, so how do we create these supercoils? First, let's get some definitions out. So there's actually something called a negative supercoil, and that's exactly what I was talking about. So if, again, you lose a twist in the DNA helix, so again, we're thinking about the DNA helix. Let me see if I can draw. So here's our DNA helix. You know how the DNA kind of twists around itself? That's a really poor job, sorry. You know how the DNA twists around itself. If I were to force one of the twists to untwist, so instead of it twisting totally, I kind of untwist it a little bit. So now it's open like that. But the rest of the DNA up here is still twisted. So let's just say that I cut it open, cut one strand open, and I forced it to untwist. So let's say we had, had a nice long piece of DNA and I untwisted one portion of it. Again, the DNA does not like to be untwisted. The DNA is supposed to have the double helix structure. And if I were to disrupt the double helix structure, what the DNA will do is create what is called a negative supercoil, which is basically the entire structure coils around itself to compensate for the fact that we're losing the twist in the helix itself. Okay, so that's obviously not good. And so that's what creates these supercoils in the DNA. Now, in bacteria, it's very important that the coils, supercoils, and everything is regulated, because we don't want the DNA to be overcoiled or undercoiled. Everything has to be perfect. And so there are these enzymes called the type 1 topoisomerase, or you can just call them topoisomerase number 1. These guys help with getting rid of supercoils. So let's say that there's too many. Here is a piece of DNA that has 5 supercoils. So that's a lot, maybe. And so if the bacteria decide, okay, we need to get rid of one of the supercoils, what the topoisomerase does is it goes into one of the DNA strands. Notice that we're zooming in. Okay, we're not cutting this. We're zooming in on the DNA double helix. So this is one strand of the DNA and the other strand. So we're only looking at this little tiny segment here. Okay, we're not looking at these two strands. We're looking at these two tiny little pieces. Okay, and what they're going to do is they're going to break open one strand. Okay, they're going to break open, it looks like the silver strand here. And then we're going to basically reestablish the helix twist. So remember here, I was saying, here's a helix. I'm going to remove the helix twist. So basically what they're doing here is this is the loss of the helix twist. We're going to reestablish the helix twist. As you can see, it looks like a normal piece of DNA now. And because we've done that, the DNA is now slightly more happy. And so instead of having 5 supercoils, it now only has 4 negative supercoils. So again, a negative supercoil is a way for the DNA to compensate for the fact that it's lost a helix twist. So in this case, it has lost 5 helix twists, but the topoisomerase will come and reestablish one of the helix twists. And so then now we only need 4 negative supercoils because now the DNA is slightly more happy. And again, I'm using the word happy just because that's just an easier way to explain it. Obviously, the DNA doesn't have any feelings, but that's how I'm going to explain it. Okay, so we went from 5 negative supercoils to 4 negative supercoils because the topoisomerase number 1 reestablished one of those helix twists. So the DNA now doesn't have that much stress on it. Okay, then there's topoisomerase 2 or type 2 topoisomerases. And there's a different mechanism for them. And these guys are primarily used to create supercoils. So the topoisomerase number 1 was used to remove supercoils, and this one is used to create a supercoil. And so what this guy does, so this is just an example, by the way, or gyrase is just an example of one type of type 2 topoisomerase. So gyre B is the first protein it's going to bind to your DNA. That's all it does. And then gyre A will come and break open one of the strands. Now, this isn't a single strand break. This is a double stranded break, meaning that it completely cuts the whole DNA off. So let me go back over here. So here, we have our DNA strands, right? You know how you have the double helix. So the topoisomerase 1 only breaks one. Okay, there's only one cut. Okay, but for topoisomerase 2, if you have, a strand of DNA here, it breaks both strands. Okay, so it basically completely breaks open the DNA rather than just one strand. Both strands are broken. Okay. So that's what gyre A does. It completely opens up the chromosome. So now it's linear essentially. And then gyre A will use some ATP to basically pass one of the strands over each other, which creates this supercoil. Basically what it does is it twists it. Okay, so it causes a twist in the DNA, and then it seals it. It reseals the DNA, so now that twist is permanent, and it creates one negative supercoil. Okay. So some things that you need to know is topoisomerase number 1 can only break one DNA strand, whereas topoisomerase 2 breaks both DNA strands, so it completely breaks the DNA open. And topoisomerase 1 is used to remove a negative supercoil, whereas type 2 topoisomerase is used to create a negative supercoil. Okay, cool. And there is a video that I'm going to post on Canvas that not McGraw-Hill, sorry. What is this book? The publisher of this book, Norton, they posted this video on how all of this works. So I definitely recommend watching that video because it's nicer when you actually see the strands moving and everything. So yeah. Okay, moving on. 7.3 is on DNA replication. And so like other organisms, DNA replication in bacteria is semi-conservative, meaning that you're going to end up with two pieces of DNA that have an old strand and a new strand, and then another old strand and new strand. So two pieces of DNA that are half and half. One thing that is unique about bacterial DNA replication is that there are two replication forks. So instead of replication just happening in one direction, it's happening in both directions. And that's again because the bacteria has a circular chromosome. So if it happens in both directions, they will meet in the middle, and DNA replication happens twice as fast, essentially. And just like all other DNA replication that you've probably studied before, DNA polymerase, which is the enzyme that does DNA replication, it can only add to the 5' end. So the direction is going to be 5' to 3'. So we'll talk about that. Okay. So just a quick overview. So the first step in DNA replication for bacteria is that we're going to create the replication forks, and this is going to happen at the ori seat, also known as the origin. So the origin or the beginning. Okay. And what's going to happen is we're going to create a bubble, because basically what's going to happen is the DNA strands are going to split at that region, and it's going to create a little bubble, and then we're going to have replication happening in both directions. So going in this direction and also going in this direction, and then eventually they'll meet in the middle. And of course, the leading strands go 5' to 3'. So we have a leading strand going this way, the leading strand going this way. But we also have the lagging strand going here and the lagging strand going there. And it's kind of funny because at the very beginning, the forks are so close to each other that the leading and lagging, this is the leading strand of this fork, and this is the lagging strand of this fork, and this is the leading strand of this fork, and this is the lagging strand of this fork. But notice that they're almost touching each other because they have started at almost the same spot. So, but you will still have a leading and a lagging strand for both replication forks. Okay, and the forks will eventually, expand and move further and further away from each other. Again, notice that we have a leading strand and all of the lagging strands, the leading strand and all of the lagging strands. So I guess I could circle, like if we were just focusing on this, right? This is what you're used to when you're learning DNA replication. So we have our leading strand, which is going into the replication fork, and then our lagging strands, which are moving away from the replication fork. But since we have two, we have a second replication fork with our leading strand going into the replication fork, and then our three little Okazaki fragments, our lagging strands, are going away from the fork. But again, because it's going in both directions, it kind of connects nicely. So it's almost always connected, except for obviously the little pieces that need to be fixed later. But those are the two replication forks. Okay, and these two replication forks get bigger and bigger and bigger, and then eventually replication is done. And the replication will finish when both forks reach the terminus region. Okay. All right, so let's actually go into the nitty-gritty. And I also posted a video, so the Norton publisher created a video on all of this. And so I got the video, and I'm going to post that on Canvas as well. So I highly recommend watching that one, because it does a really good job of showing like all the enzymes moving and how replication happens. So DNA replication in bacteria obviously is still slightly different from DNA replication that you've probably learned with eukaryotes. So we're going to go through all of the steps. So the first step in DNA replication is initiation, meaning that you have to begin, right? And so we have this protein called DNA A. The way I remember it is A is the first letter in the alphabet. And you're going to see why, because later we have DNA B and C. So A is first, essentially. So DNA A will bind to the ORI site, the origin of replication. And it binds to these specific sequences that have like 9 DNA base pairs. But anyway, it doesn't really matter. All you really need to know is that the DNA A binds to the ORI C site. And what DNA A does is, as you can see, this is the DNA. And it makes the DNA fold over, okay? It forces the DNA to, instead of being like a nice flat line here, it forces the DNA to fold in on itself. And when the DNA folds in on itself, it creates this opening right here. So notice that the DNA is nice and double-stranded and everything looks beautiful, but as soon as we force it to flip over like this, it causes these two strands to break open, because again, DNA is not meant to be bent that tightly, and so it forces the two strands to open here. And now that we've opened up the two strands, now we can add all of our enzymes that are required for DNA replication. So the first one is going to be the DNA helicase. So DNA helicase, which is DNA B, helicase and DNA B are the same thing, are going to come, and it's going to, the helicase, it also comes with this DNA C. DNA C is what we call the loader. All it does is it helps load the helicase onto the DNA, basically. So DNA C is our loader, and it loads the DNA B, which is our helicase, onto the DNA strand, okay? The loader protein will just leave, okay? So the blue guy is the loader, and the loader just leaves, okay? So now we have 2 DNA helicase proteins attached. Why do we have 2? Again, remember DNA replication in bacteria happen in both directions. So we're going to have a helicase going in one side and the helicase going on the other side. Okay, fantastic. And so now the helicase can do its job and start unwinding the DNA. So it'll go this way and it will go that way, and we're going to be unwinding the DNA. Okay. Still going. So after we've unwound the DNA a little bit, now there's some space, and so DNA primase can come and add, I should say RNA primers, sorry. Gonna come and add RNA primers. Okay. And yeah, RNA primers are gonna get added. Remember that DNA polymerase needs these primers to be able to start the DNA replication. So the primers are important. So now that the primers are there, we can add the DNA polymerase. And the way that it comes is through these clamps. So it's like really freaking crazy how it looks, but there's this giant structure called a clamp loader, okay? And it's carrying the DNA polymerase, actually 2 of them. 2 DNA polymerase attached to this giant clamp loader. And then inside of that is a sliding clamp, which is I guess the thing that helps slide along the DNA strands. Okay, and so this whole structure will come and it will attach itself to both strands. So as you can see, one on the top strand and one on the bottom strand, again, because we need to go in both directions. So everything is happening double, basically. Okay, so the DNA polymerase comes via this clamp / sliding clamp structure. And by the way, as this is happening, the helicase is still going, right? It's not like the helicase stopped. So the helicase is still unwinding the DNA. And now we are adding, because now we have enough space, we also add the primer for the lagging strand. Okay, so the lagging strand receives its primer as well. So the leading strand gets its primer first, and then the DNA polymerase comes, and as that's happening, the helicase is unwinding, and now we have space to create the lagging strand primers as well. Okay, so now that we have both primers, leading strand primer and lagging strand primer, we're going to start actual DNA replication, okay? And so the leading strand is the one that first goes. So the leading strand is the one that begins. The lagging strand is lagging, so it's going to take a second. But the leading strands begin polymerizing or being replicated almost immediately. Okay, so that's initiation. Then we have actual elongation. So we're just going to keep on going, right? So as the helicase is unwinding the DNA, the DNA there's a huge segment of DNA that is single-stranded. So there are these single-stranded DNA binding proteins that come, and they have to attach to the single-stranded DNA to protect it. So we can't have single-stranded DNA just kind of out and about. So this SSB binds to the single-stranded DNA. Okay, whatever. That's not really the most important part. The most important part is really what's happening with the lagging strand. So the lagging strand is now being elongated. Notice that we don't really care about the leading strand. The leading strand, once it begins elongation, that's it, right? It's just going to go on forever, and nobody really cares. It just continues. The DNA polymerase goes. There's nothing special about it. It's really the lagging strand that is the issue. And so the lagging strand is going to be elongated, as you can see. So we're elongating the lagging strand, and the lagging strand is being elongated. And basically every 1,000 bases, a new lagging strand primer is made. So as this is happening, every 1,000 bases that the whole fork is moving, a new lagging strand primer is made, because again, the lagging strand has to be made in fragments, right? Okay. And then the lagging strand will eventually hit the previous fragment, right? So here's your lagging strand. It's filling in this way. It's going in this direction. It's going this direction. And then as it keeps going, it will hit this part of the DNA, which has already been replicated. So then it will come off and fall off. Okay. All right. And so when it falls off, then we have to reestablish the clamp. And so the clamp basically grabs another DNA polymerase, and it will attach to the new RNA primer. Remember how we just created a new RNA primer? And so then the replication continues, and then it will keep going again, and then it will stop over here, and then the whole thing just repeats over and over again. So the Okazaki fragments are made, at, I guess, 1,000 base pairs at a time. And this just is happening over and over again. So the clamp never really comes off. Okay, the clamp just receives a new DNA polymerase every time we create a new Okazaki fragment. Okay, cool. So like I said, there is a video that I posted about how this all works. And I feel like the video does a really good job because you can see everything moving. So I highly recommend watching that. Alrighty. As you probably know, because we have all of these little tiny pieces of RNA primer, we have to get rid of that RNA primer. And so RNase H is the enzyme that bacteria use. It removes the RNA primer, so it chops it up. And so now there's a big gap. And so then we have DNA polymerase number 1 comes in and fills that gap, but it doesn't fill it all the way because it can't fix this last little piece. There's just a tiny little piece of the DNA that the DNA polymerase 1 just cannot fill. It just cannot make this covalent bond. And so then we have a third enzyme, DNA ligase, which comes and repairs this nick, basically, which is a small little cut in the DNA. So DNA ligase comes and it seals the nick, and so now it's a continuous piece of DNA. All right, so that's all that's happening during DNA replication. Now, how does it stop? Right, so remember, DNA replication is happening in this way and the other way. So it's happening in both directions. So how do we get it to stop? Well, at the end of replication, there are going to be these ter sites, the termination sites, at the very end. Basically, they're opposite of the origin. So if the origin is here, the ter sites are basically on the complete opposite of the chromosome. And basically, once the replication fork gets here, it tells the forks to stop, and DNA replication can end. Now, so that's this part A here. This happens all the time. Because the plasmids are circular, after DNA replication, they almost always are linked like this. So they're linked chromosomes, really annoying. And obviously that's problematic because if we're trying to do DNA, if we're trying to do cell division, there's no way that these two are going to separate. So that's not good. So another enzyme, topoisomerase 4, comes, and it's going to come in, break open one of these, and unlink them. Basically, it just breaks one open and then unlinks it. So two link together, the topoisomerase opens one, closes it, and so they're unlinked. That's pretty much it. Topoisomerase number 4 is very similar to topoisomerase number 2 in the sense that it can break 2 strands open, not just one. So it is basically a type 2 topoisomerase. Yeah, the way I remember it is that 4 and 2 are both even numbers, so they're both type 2 topoisomerases. Okie dokie. So that was DNA replication in bacteria, kind of in a very short, short video. So again, I will post on Canvas the publisher's videos on all of this, so you can watch those, because I do recommend. I really like how the animations move and everything. Okay. So now let's move on to 7.4, which talks about plasmids. So plasmids, again, are these small circular pieces of DNA. And why are these plasmids important? Well, again, they carry useful but non-essential genes. So what that means is that the genes are useful, meaning, ooh, maybe it gives a bacteria like a new characteristic or something like a survival gene, but it's not essential, meaning that it's not like a gene that is necessary for the growth or metabolism of the bacteria. It's just like a gene that would be nice and is helpful, but it's not necessary for like living. Okay. Some bacteria can have many copies of one type of plasmid. So plasmids can come in one copy or many copies. So that's cool. And then the plasmids, the most interesting thing is that plasmids, because they're such small pieces of DNA, bacteria can pass it to each other. So a plasmid can move from one bacteria to another, which is very interesting because now bacteria can share DNA and genes through plasmids. Okay. So plasmids replicate as well because, again, when a bacteria divides, it would like to keep the plasmids that it has. And so plasmids replicate as well, and they replicate completely separate from the chromosome. So we're going to talk about how plasmid replication happens, and there is also a video on this. I'm going to post it on Canvas. There's also a video on how plasmids are replicated. So the model that we call it is called the rolling circle model. And the reason why is because it doesn't happen in both directions. So you know how with the bacterial chromosome, it's a two-direction, there's two replication forks. So with the plasmid, there's only one region where the replication happens. Okay, so let's start with the first part. So here is the ori site, the origin, and this enzyme called Rep-A is going to come in and it's going to break it open. Okay, notice that it only breaks open one strand. It doesn't break open both strands. It just creates a nick where one strand opens up. Okay, cool. So that happens, and then we're going to get DNA polymerase 3 is going to come over here, and it's going to extend. That's pretty much what's going to happen. Okay. Oh, by the way, this rep dimer just kind of sticks here the entire time. But anyway, DNA polymerase is going to come here and it's going to extend the 3' end, and it's just going to keep extending it. Now, if it extends it this way, this piece of DNA, the outer, I'm going to call it the outer strand. Obviously, there's no such thing as an outer and inner strand, but in this diagram, you can clearly see that there is an inner strand and an outer strand. Okay, so we have cut open the quote-unquote outer strand, and the DNA polymerase is going to come over here, and it's going to extend from the 3' end and go around like this. Now, obviously, this strand has to get out of the way for that to happen. So, yeah, that's what we're going to do. Give me 1 second. Feel like, okay, I'm just going to, hopefully that fixed it. Okay, anyway, so, yeah. We're going to get DNA polymerase is going to come, and it's going to come and replicate the outer strand, quote unquote. Again, there's no such thing as an outer strand. So here is that happening. So the DNA polymerase is creating this new outer strand. And what's happening to the old outer strand is that it's literally peeling off, okay? And as it peels off, notice that it creates a little circle itself. So it's basically creating its own little plasmid. Okay, cool. That's nice. And then what's going to happen at the very end is that the Rep-A will cut right here. So this plasmid is now whole. Okay, it's a double-stranded plasmid, And then now we have this single-stranded plasmid, which was that old outer strand, right? The outer strand got pushed out, and now it's just by itself like this. Okay. DNA polymerase will come over here, by the way, and fix this. It will come and fill in the gaps, so now it becomes double-stranded as well. So then you end up with two plasmids that are both double-stranded, one that has an old inner strand and a new outer strand, and another one that has an old outer strand and a new inner strand. Again, there is really no such thing as an inner and an outer strand, but just because of how the diagram looks, it just makes it easier for me to explain it that way. Okay, cool. All right, moving on. So plasmids have to regulate their replication. Certain plasmids replicate all the time, so you can have like hundreds of copies of that plasmid inside of the bacteria. And some plasmids, maybe there's only one or two copies. So different plasmids regulate their replication all the time. And so how does this happen? So remember that Rep-A enzyme. So the Rep-A can produce these dimers, which are basically just two Rep-A stuck together. And they're going to bind to these regions called interons. Wait, I totally spelled that wrong. Interons. It's spelled right in the diagram. I didn't spell it this. Okay. Interons. Okay, those are regions on the Ori C. And notice that what happens when there are a bunch of these Rep-A dimers is that it basically makes two plasmids get stuck together. So we call it a handcuff, because we're handcuffing two plasmids together, like when your hands get handcuffed. And so when two plasmids are handcuffed like this, there's no way that DNA replication can happen. And so that this formation turns off replication of the plasmid. Now, if we want to turn on the replication of the plasmid, we can get rid of this complex, usually through proteases and stuff like that. But basically, we're getting rid of these dimers. And so now that the dimers are not there and the handcuff is basically broken, then we can add the helicase and the DNA replication can continue. So the helicase can begin replication here. So this is just how the plasmid regulates how much replication is happening. It will handcuff itself if it doesn't want to replicate, and it will undo the handcuff if it wants to continue replication. Okay. All right. So another issue that some plasmids have to deal with is segregating from itself. So this cell, if it were to divide, there's a chance that both plasmids end up in one daughter cell and then the other daughter cell ends up with none, right? So if you were to imagine here is a bacterial cell that's about to divide, if both of the plasmids end up here and then division happens, then you end up with one daughter cell that has two plasmids and one daughter cell that has no plasmids. Which is obviously not good. So we don't want that to happen. And so plasmids have a way to segregate each themselves to make sure that each of them are on opposite ends of the cell. And, you know, it makes sure that each of the daughter cells gets a plasmid. Very similar to regular DNA regular cell division where the chromosomes have to split. Yeah, it's kind of like that. Anyway, so here are the proteins that are involved. So we have PAR-R. PAR-R is a protein that kind of binds to the surface of the plasmids, both of them. That's really all it does. And then the PAR-R binds to PAR-M. And PAR-M is this protein that produces a filament. So a filament is just like a long strand of protein. And so notice that this plasmid is creating this, not really creating, but the PAR-M is creating a filament on this plasmid and then also on this plasmid. Okay. So now these two plasmids have these short little filaments that are kind of sticking off of it. And so now they're just kind of chilling. And then in step 3, what ends up happening is they connect to each other. So this part, the little tail end of this filament and the tail end of this filament actually attach. So now they're attached to each other through 2, technically 4, but anyway, 2 filaments. Okay, so now they're attached like this. Okay, and so what ends up happening is the filaments continue to grow. So we're down here now. The filaments grow, and it causes the plasmids to be pushed away from each other. So notice that the plasmids are, the distance between the plasmids is increasing until they reach the very ends of the cell. Okay, and when they reach the very ends of the cell, the PAR-M filament starts to disintegrate or disassemble. And that's good because now the two plasmids are already separated, so that's all they needed to do. And we just needed to push them apart, and that was pretty much it. Yeah. Okay. Secondary chromosome. So what is a secondary chromosome? It is a piece of DNA that carries at least one essential gene. So remember, I told you that a plasmid does not carry any essential genes. It just carries extra genes that maybe are useful, but are not essential for survival. So secondary chromosomes are an actual piece of DNA that is not the main chromosome, but just like a smaller piece of DNA that contains at least one essential gene, meaning that if the bacteria lost this chromosome, it would not be able to survive. And so the theory is that a plasmid can become a secondary chromosome if it acquires some kind of essential gene. So here is a bacteria with an essential gene, and let's say that it receives a plasmid, just a regular old plasmid, not anything special. So this plasmid can obtain or take in a bunch of non-essential genes, which is cool. That's what plasmids do. But there are a couple ways where it can possibly become a secondary chromosome. One way is if the essential gene from the main chromosome translocates onto the plasmid. So just a transfer. Obviously, that makes it so that this is now a secondary chromosome. Okay. Another way is if the plasmid acquires some kind of non-essential gene from the outside, some external source. So that's through horizontal gene transfer, which we'll talk about later in the class, but basically a gene from outside goes into the bacteria and then attaches itself into this plasmid. Now, this plasmid has this essential gene, but there's actually two copies of that essential gene. So if for whatever reason, the essential gene in the main chromosome gets deleted, now the secondary chromosome is the one that carries that essential gene, and it's the only one that carries it. And so now this is considered a secondary chromosome because without this, the bacteria would not be able to survive. Alrighty. So now we're moving on to 7.5. We're going to talk about just really quickly eukaryotic and archaeal chromosomes. So with eukaryotic chromosomes, you probably already know this because this is kind of the main type of chromosomes that we talk about in like cell bio and stuff. But eukaryotic chromosomes are packaged with histones. They do have introns, which is annoying. And then they also have a lot of non-coding regions, which we saw earlier in the chapter. Now with archaea, their chromosomes are a little bit interesting. They do have single circular DNA, just like bacteria, and they do go through bidirectional replication, meaning that there's two replication forks. But they do have multiple origins of replication. So unlike bacteria, there's multiple origins. Most people don't actually know why there are multiple origins, but they do. So that's something that makes them interesting. Okay. Last thing, oops, that didn't show up. Okay. Last thing we're going to talk about is the metagenome. So I kind of just like briefly went over this, but metagenomes, as we've already talked about, is a way to sequence all of the genes in a single sample. So we're not sequencing just one organism, we're sequencing all organisms in a sample. And it is very useful for determining what kind of species lives in an environment. So here in this diagram, basically some person took samples of a bunch of organisms in different places in the ocean, and then they determined like what types of bacteria are living in certain parts, which is cool. They can also be used to monitor the genes in an environment. So I know some people who study like how many antibiotic resistance genes there are, in the ocean versus the rivers and, things like that. So it's just a nice way to look at the bigger picture rather than like a small thing. Okay, cool. Thanks for watching, and I will see you in the next video. And please remember to watch those supplementary videos that I'm going to post on Canvas. All right.